Iron is a vital element for oxygen transport, DNA synthesis, and cellular energy production. The human body has no active excretory mechanism for iron, making the regulation of its absorption from the diet the critical control point for maintaining balance. We begin with dietary sources, distinguishing between two key forms. Heme iron is found within hemoglobin and myoglobin from animal products such as red meat, poultry, and fish. It is highly bioavailable, with absorption rates between 15 and 35%. Non-heme iron is found in plant-based foods like lentils, beans, spinach, and fortified cereals, as well as in iron supplements. It has lower bioavailability, typically between 2 and 20% absorption, which is significantly influenced by other dietary factors. The primary site for iron absorption is the duodenum and proximal jejunum. The process differs for the two iron forms. For non-heme iron, the first step occurs in the intestinal lumen where ferric iron is reduced to ferrous iron by an enzyme called duodenyl cytochrome B located on the brush border of the enterocyte. This ferrous iron is then transported across the apical membrane into the enterocyte by a protein called divalent metal transporter 1. For heme iron, the process is different. Heme is taken up intact across the apical membrane via the heme carrier protein 1. Inside the enterocyte, the enzyme heme oxygenase releases ferrous iron from the porphyrin ring of the heme molecule. Once inside the enterocyte, regardless of source, the iron faces a choice. It can be stored within the cell as ferritin, and subsequently lost when the enterocyte is naturally shed at the end of its life cycle, or it can be transported out into the bloodstream for systemic use. The transport of iron into the systemic circulation is a carefully controlled process. Inside the enterocyte, ferrous iron must be oxidized to ferric iron. This is facilitated by the protein hepcidin. The ferric iron is then exported across the basolateral membrane of the cell by the sole iron exporter, a protein named ferroportin. Once in the plasma, ferric iron binds tightly to a transport protein called transferrin. Transferrin is the major iron transport protein in the blood, and under normal conditions, it is about 1/3 saturated with iron. Transferrin-bound iron is delivered to cells throughout the body that express transferrin receptors, with the majority going to bone marrow erythroid precursors for the vital synthesis of hemoglobin. Cellular uptake and utilization occur through a highly efficient process known as the transferrin cycle. Cells with high iron demand, such as developing erythroblasts in the bone marrow, express high levels of transferrin receptor 1 on their surface. Deferric transferrin, carrying 2 iron atoms, binds to this receptor. The entire complex is then internalized into the cell via a process called receptor-mediated endocytosis, forming an endosome. Within the acidic environment of the endosome, iron is released from transferrin. It is reduced back to ferrous iron and transported out of the endosome into the cell's cytoplasm by divalent metal transporter 1. The iron is then utilized for essential cellular functions. In erythroid cells, it is transported into mitochondria for incorporation into heme as part of hemoglobin. The now empty transferrin, called apotransferrin, and the transferrin receptor are both recycled back to the cell's surface to be used again. Not all iron is used immediately. The body maintains significant storage reserves. The primary storage form is ferritin. Ferritin is a spherical, soluble protein complex that can store up to 4,500 iron atoms in a non-toxic ferric oxyhydroxide form. It is present in almost all cells, with the highest concentrations in liver hepatocytes and macrophages of the reticuloendothelial system. Hemosiderin is an insoluble, aggregated degradation product of ferritin that accumulates in lysosomes, particularly when iron stores are very high. Iron in hemosiderin is less readily available for mobilization than iron stored in ferritin. Clinically, the serum ferritin level is a crucial laboratory indicator of total body iron stores, with low levels indicating depletion and high levels suggesting overload or inflammation. Remarkably, the majority of the body's daily iron requirement is met through recycling, not dietary absorption. This recycling is performed by the reticuloendothelial system. Senescent red blood cells, At the end of their 120-day lifespan, are phagocytosed by macrophages, primarily in the spleen and liver. Within these macrophages, heme from the degraded hemoglobin is broken down by the enzyme heme oxygenase, releasing iron. This iron can then be stored within the macrophage as ferritin, or more importantly, exported back into the plasma via ferroportin to rejoin the transferrin pool for immediate reuse, particularly for new red blood cell production. The entire system is governed by a master regulatory axis, the hepcidin-ferroportin system. Hepcidin is a small peptide hormone synthesized in the liver. It acts as the master regulator of systemic iron homeostasis. Its mechanism of action is direct. Hepcidin circulates and binds to the ferroportin protein on the surfaces of key cells like duodenal enterocytes and reticuloendothelial macrophages. This binding causes ferroportin to be internalized and degraded, effectively locking iron inside these cells and preventing its export into the blood plasma. Several key stimuli regulate hepcidin synthesis. Increased body iron stores, signaled by high serum ferritin and high transferrin saturation, stimulate hepcidin production to prevent further absorption. Inflammation and infection, particularly through the cytokine interleukin-6, strongly upregulate hepcidin, leading to the characteristic iron trapping seen in anemia of chronic disease. Conversely, hepcidin synthesis is inhibited by erythropoietic demand. When the bone marrow needs more iron to make red blood cells, such as during anemia or hypoxia, it produces a hormone called erythroferrone, which suppresses hepcidin. Iron deficiency itself also directly signals the liver to reduce hepcidin production, allowing for increased dietary iron absorption and macrophage iron release. Understanding these pathways explains common pathophysiological conditions. In iron deficiency anemia, caused by inadequate intake, malabsorption, or chronic blood loss, the body's iron stores become depleted, reflected by low serum ferritin. This leads to low hepcidin levels, which maximizes iron absorption and release, but the demand still outstrips supply. In anemia of chronic disease, also called anemia of inflammation, the underlying condition drives high levels of interleukin-6, which elevates hepcidin. The high hepcidin traps iron within macrophages, making it unavailable for red blood cell production despite the body having normal or even increased total iron stores. In hereditary hemochromatosis, a common genetic mutation often in the HFE gene results in inappropriately low production of hepcidin. Without adequate hepcidin, ferroportin on enterocytes and macrophages remains unchecked, leading to excessive and uncontrolled iron absorption from the diet, resulting in toxic iron deposition in organs like the liver, heart, and pancreas. Thank you for watching. Don't forget to like, share, and subscribe. See you in the next one.